Hello, can anyone hear me? All right, can someone hear me? I'm going to assume you guys are hearing me fine. My volume seems to be
Yes, I can hear you.
okay there. You turn up the volume on. Hmm. All right, we're a little behind schedule, I think. Today's December 7th, and let me take that back. We're on schedule. We need to finish chapter 13 today. Viruses, viroids, and prions. If I do not finish it, maybe I will give it in the lab, but I hope to finish today. Last time I had trouble with Zoom. Let me see if I got this going. Everything's good. There was no recording that I could put up, so I put up a recording from an earlier term. Are there any questions before we begin?
Nope.
All right. So let me begin. I'm going to cover this slide again because it wasn't in the video and it was the only slide that was not in the video. Uh, there are some cancers which are caused by viruses. The exact mechanism of how a virus causes a cancer is not totally understood. And there's more than one change that's needed to get a cancer, but you don't need to know all of that. All you need to know is that some cancers, about 10 to 15% of human cancers, appear to be caused by viruses. It's well known that human papillomaviruses cause both cervical and skin cancer, and hepatitis B virus and hepatitis C virus cause liver cancer. Not all cancers are caused by viruses. For example, breast cancer and glioblastoma appear not to have any association with viruses. Any question about any of that? All right. Cancer, all you need to know is it's uncontrolled growth of a person's cells. You don't need to talk about anything else about the cancer, but you should just know that cancer is uncontrolled growth. It's your own cells, by the way, cancer. There are two main ways that viruses can cause cancer. First, a virus can integrate in the DNA, and in eukaryotes, we call that a provirus. So in humans, it would be a provirus. And then the cancer genome becomes a permanent member of that cell's DNA, causing a permanent genetic change in the cell, including if the virus integrates inside a gene, you've obviously knocked out that gene. You can change the expression of genes and other aspects, meaning a virus can move a promoter, which gets transcribed, and then you can turn on a later gene lower down in the human genome. So the formation of proviruses just mucks up the DNA, creating permanent genetic changes in cells. And obviously, this can cause a cancer. I think I did mention that retroviruses appear to integrate essentially at random in different cells, and they can change the genetic makeup of these cells. And that can be dangerous when a person gets a retrovirus because they have DNA integrating in different locations in different cells. And one of those cells could become mutated and then go on and form a cancer. That's the first way that viruses can cause cancer. There is a second way, and that is an oncogenic virus can enter a cell, and an oncogenic virus contains an oncogene. An oncogene is a gene involved in normal cellular growth and regulation. However, the oncogene is mutated, and then the cell just grows. So it doesn't undergo normal cell growth and regulation. It just grows because it's a mutated gene. And mutated oncogenes help transform normal cells into cancerous cells because the cell gets the oncogene and then the cell just grows. Further changes in the cell can lead to cancer. This is an advantage for the virus because the virus then infects a cell, which the cell then grows, and that makes more viruses. All right, any questions about how viruses can cause cancer? They can do it by simply integrating at random, making a provirus, changing the genetic makeup, or the virus can bring in an oncogene, which makes the cell grow. Any question about any of that? All right, we mentioned that viruses can be lytic in animal viruses. They can be a lytic virus. They can also be a budding virus. Well, it turns out that some eukaryotic or animal viruses are actually latent. These are usually budding viruses that stop the budding life cycle and then go into latency, where the virus goes latent. The virus remains asymptomatic, doesn't cause disease, and it can remain in the host cell for long periods. Unfortunately, this virus can leave latency and then become active again. And usually, like I said, it'll enter the budding life cycle. This happens with cold sores. While the virus is active in the budding life cycle, where the virus is metabolically active and reproducing, you get the cold sore. And then you get better, and you might not get a cold sore for a year or several years later. And that's because the virus went latent, it's inactive, not causing disease, and then it comes out again in the budding life cycle, causing the cold sore. Herpes simplex virus 1 does that. Another example is the chickenpox virus. While it's metabolically active, infects a child, or usually a child, causing chickenpox, and the child is sick with chickenpox, and then the virus leaves the budding life cycle and goes latent. And this virus can go latent for decades. And then it will leave latency and re-enter the budding life cycle, causing shingles. This virus, the first time it infects a person, will cause chickenpox. All other times the virus becomes active, meaning it leaves latency, it will cause shingles. Usually most patients only get shingles once, but you can get shingles multiple times. I knew a lady who was a friend of a friend, and she got shingles about every year. There's usually a trigger for making the budding virus leave the latent life cycle and then re-enter budding. With a cold sore, what it is it's usually just the person gets a cold and then they get the cold sore. With shingles or chicken pox virus, whatever that virus is. The latency is usually old age, where the immune system weakens, and then the virus becomes active again, causing shingles. And that's why usually when a patient gets this virus, it's in childhood and they get chickenpox, and usually they're in old age when they get shingles. It doesn't have to get that way. For example, there was uh, a fetus who was exposed to chickenpox when the mother got it. And then the baby, shortly after it was born, it was still a baby. So in the first year of life, the baby got shingles. Any question for any of that? All right, like I said, there's usually a trigger, which is a stress, which makes the virus leave latency and then usually it enters the budding life cycle. So in animal cells, there's the lytic life cycle, some viruses can follow, the budding life cycle, and the latent life cycle. There's one other, and that is the persistent viral infection. This is a virus that is a naked virus, but it tends to follow the budding life cycle in that the virus, when it replicates, it does not replicate at large numbers, so it does not kill the host cell. It's a naked virus, but it's shed by the virus is put in a vesicle, and then the vesicle comes up to the cell membrane, dumping the contents out of the cell. And so I don't know, it's not exactly budding, but it's produced at low numbers, and it's not a latent virus, a naked virus. And it's put in a vesicle, and then the vesicle moves to the cell membrane, merges with it, and dumps its contents out of the cell. A persistent virus can cause a persistent viral infection. And I'm sorry, the two words mean different things. There's the persistent viral life cycle, and then the persistent viral infection. A persistent viral infection is a viral disease that usually processes slowly over a long period of time, and it may be caused by a persistent viral life cycle. It doesn't have to be. The detectable virus load increases over a long period of time. Oftentimes, the virus is never cleared by the host immune system, and it may be fatal. AIDS, before the modern medication, was an example where HIV infected the patient. It would be a persistent viral infection. It would get worse over time. The patient would eventually develop AIDS, and then they would die. Obviously, with modern medication, that may not be the case. The patient, though, would get a persistent viral infection because most of these patients will not clear HIV from their immune system, even with medical treatment, meaning they can live close to a normal lifespan. It's a little shorter, but it's close to a normal lifespan with modern medication. But the virus is usually never cleared. Some of these persistent viral infections are tolerated, like cytomegalovirus. Some people can be infected with cytomegalovirus, and they will consistently shed it, but it grows over time, getting worse. I'm trying to illustrate that, but it plateaus and then stops getting worse, and then it just continues at that level. Chickenpox actually is a persistent viral infection where first you get chickenpox, then it enters the latent life cycle, and then it can come out as the budding life cycle again and giving rise to shingles. It can occasionally enter the persistent viral infection. My mother got this when she got shingles. She had shingles for the rest of her life. And so she had shingles for about 3 years. It never went away. There are some other herpes viruses which are tolerated and give a persistent viral infection. Any question about any of that? Some people call mononucleosis a persistent viral infection. And that's because it does go away, this one, but it does tend to grow slowly, meaning the viral load starts slowly over time. And then it just keeps growing. And maybe it'll bloom, meaning it'll expand like that. But with this one, it does reach a plateau and then it drops away. And then you actually are cured of mononucleosis eventually, but meaning it'll drop to zero. But it takes a long time. And in this process, for when you have the symptoms, the mono lasts for several months usually. It doesn't get better for a long time. Any question about any of that? So with viral infections, you can have an acute viral infection, and that's when the patient first gets infected. They quickly get a high load of virus, like the common cold, and then the virus drops very quickly and then goes away. An acute viral infection, common cold. The flu actually does follow that too. It's just that it's much more severe and it does last longer than the common cold. But it is an acute viral infection. There's the latent viral infection, and that would be the child first gets chickenpox, and that would be like an acute infection. And then the chickenpox goes away, but the virus does not. It's staying dormant in a latent life cycle in the ganglia of your nerves. And then much later, the latent infection happens where the virus leaves latency, enters the budding life cycle, and the patient gets shingles. It doesn't have to be that long. Like usually chickenpox happens in childhood and shingles most of the time happens in elderly lifespan. But like I said, it doesn't have to happen that way. And then there's a persistent viral infection, which I already told you. The patient gets infected around here. The viral load grows slowly. It may bloom. And you see the graph is broken here. That's because in this case, the patient dies, like HIV causing AIDS, and then the patient dies. It doesn't have to. It can be tolerated, meaning it can bloom to this stage and then continue that way. Like that's usually how cytomegalovirus goes. Any questions about the different viral infections?
Nope.
All right. If not, let's move to prions. Prions are an infectious protein. So a proteinaceous infectious particle. And they cause disease. And this is the only known case where proteins are infectious, meaning they can spread from one patient to another, although the spread isn't very easy. You have to usually consume those proteins. Although I imagine there is some other way it spreads because scabies is a disease caused by prions that have been plaguing sheep farmers in Europe for centuries. And so I'm imagining that scabies shed some way and then infect other sheep another way. It might be that protein is shed some way and then gets on the grass and then the sheep eats it. I don't know. I don't know how scabies spreads in sheep, and I don't know if anyone knows. But they are inherited, transmissible by ingestion, transplant, like you can get prion from an organ transplant, and by surgical instruments. Prions cause spongiform encephalopathy. These are vacuoles in the brain, sorry, in humans and other animals. Let me show you a picture of that. You see why it's called spongiform. Let me blow this up. The first is what a normal human brain looks like. This is a prion-infected brain. This is a prion-infected brain. And that's a prion-infected brain. And you can notice these sort of look like sponges. Why it's called spongiform. Encephalopathy has to do with the brain. Let me end this. In animals, the most common example is bovine spongiform encephalopathy, more commonly called mad cow disease. But it also causes scabies in sheep. And this one is a disease, like I said, we've known about for centuries. European sheep ranchers have been living with this disease for centuries. But it does cause other disease like cervid, chronic wasting disease, or recently in Colorado, it's fairly recently in the last 20 years, they found, I think it's elk, get it, and I'm just going to call it elk uh, spongiform encephalopathy. In humans, we don't call it that or any of those. We call it Creutzfeldt-Jakob disease after two doctors who discovered it in patients, or the variant of that, Creutzfeldt-Jakob disease, or Gerstmann-Straussler-Schnickler syndrome. And I could be butchering that name. I'm not German, and I don't know how to speak German. Followed in certain families, and the diseases were a little different. I'll explain that in a minute. And then fatal familial insomnia, and then Kuru. Kuru is the one I understand the best, but these two, Creutzfeldt-Jakob disease and Gerstmann-Straussler Schnickler syndrome, are actually diseases that were running in human families, so that people are born with the genetic predisposition to getting the disease, the spongiform disease. And what happens is they have a protein which can easily change into the prion protein. And we'll talk a little bit more about that. Any questions about any of that? All right, so we don't call it spongiform encephalopathy in humans. I don't know why, but we call it Creutzfeldt-Jakob disease. And you do see the spongiform encephalopathy, but they call it that. And I'm not going to try saying that one. Or Kuru. Let me talk a little bit more about Kuru, because this one is understood a little better than the others. Kuru is a disease that was seen in the Fore tribe in Papua New Guinea. This is Papua New Guinea, the island above Australia. And I think Papua New Guinea is only this part of the island. And let me blow that up. The red place is where the Fore tribe was. And when the patients in the Fore tribe got Kuru, they were unable to walk, lost the ability to swallow or chew, had drastic weight loss, which led eventually to death. How was Kuru transmitted? The Fore tribe had an unusual funeral ritual where they cooked their dead and then ate their dead relatives. And when they ate the nervous tissue of the relatives, they ate the prions, and then that gave rise to Kuru. Fortunately, the mothers of the Fore tribe made the connection that when they ate their dead relatives, and then fed it to themselves and their children started coming down with Kuru. And so the mothers themselves stopped feeding their dead relatives to the children, and they also stopped eating the dead relatives. So when the missionaries came to Papua New Guinea, they found that only the men were cooking their dead relatives and eating them, and essentially only the men were coming down with Kuru. And the missionary said, "This is anathema. We can't allow this." And they stamped out the funeral ritual, meaning there's no more eating their dead relatives. That actually got rid of Kuru in the Fore tribe. And you can say all you want about missionaries and how they're bad for the culture. In my opinion, and this is my opinion, the missionaries did the Fore tribe a favor. They got rid of Kuru. Any question about any of that? All right. How do prions, an infectious protein particle, spread? How do they cause disease? Well, in normal cells, there is a normal protein, and it's important in the cell surface of nerve cells, and it's called PRP superscript C, the normal protein made in the nerve cell. This normal protein can come up to the prion protein, which is PRP superscript SC, SC for Scrapie's protein. I'm just going to call it the prion protein. And these two proteins have the same amino acid sequence. So what's the difference between these two proteins? They have a different folding pattern. So the protein is folded in a different pattern. And the Scrapie's protein, or the prion protein, can come up to the normal protein and then change the folding pattern of the normal protein to make it a prion protein. Now this cell has two Scrapie's or prion proteins, and it doesn't have enough PRPC protein. So the cell says, oh, I need to make more PRPC protein because the cell needs it for its functioning. And now there's more PRPC protein. The trouble is the cell has two prion proteins. And if it comes up to the normal protein, it'll induce it to change its folding pattern into the Scrapie's protein. And then the cell will make more PRPC protein, and then more of it will be changed into the prion protein. The prion protein accumulates in the brain cell, start forming plaques, and eventually will cause cell death. And that's how the prions cause disease and how the prion protein replicates. Any question about any of that? The surprising part is that the two proteins are the same amino acid, they're just in a different folding pattern. As I mentioned, the people who get Creutzfeldt-Jakob disease in their family, they are born with a PRPC protein that has a genetic makeup that it is more likely to change into the prion protein. So normally what happens is their cells make this protein, and every once in a while, one of their PRPC protein spontaneously changes shape into the prion protein. And then the prion disease starts. Usually in these families, the prion disease does not happen until the patient is at least in their middle ages. And that's why the families are continuing, because when they're young adults, they're reproducing. And then there's children born in the family who later on will develop prion disease, the Creutzfeldt-Jakob disease. Any question about any of that? All right, I think that's it for prions. Oh, let me state that we did have an epidemic of prion disease. I'm sure you all heard about it, mad cow disease in England. How it was spread was the British wanted some use for cow brains because they couldn't sell it to the public. Nobody wanted to buy cow brains and eat it. And so what they did was they took the cow brains and ground them up and then added the cow brains to animal feed, meaning grain. And then they fed that grain to cows. So the cows, when they ate the grain, were essentially eating cow brains with it. Well, one of the cows became infected, probably spontaneously, or maybe it was first infected with scrapies, meaning maybe they put sheep brains on the grain, sheep brains in the grain, and then the cow developed mad cow disease. And then they took that cow when it died and took the brains and put it on to feed. And you can see how that would spread mad cow disease among more cows. Excuse me. And then those cows, their brains were taken and added to the animal feed. And it just bloomed out of proportion. The British tried to control it by checking the cows and killing and incinerating any of the cows that came down with mad cow disease, but it was a impossible task because they continued to take cow brains and add it to their grain, and so more and more cows just came down with mad cow disease, even though they were essentially removing any of the cows that develop mad cow disease. The trouble is the cow can have the prion for a while before they ever have disease or show signs of disease. And I don't remember how long the period is, but it's really late in the disease process before the cow comes down with mad cow disease. And so the British had to actually incinerate all of their cattle and then import new cattle from outside Britain to get rid of the mad cow disease. I can state that more than cows came down with the disease, the prion disease, although we didn't call it mad cow disease. There were about 150 British people who came down with the prion disease, probably from, or most certainly from, eating beef tainted with mad cow disease. And there were also 150, close to that, British cats, both domestic kitty cats and zoological cats, that came down with mad cow disease. So cats were very susceptible as well as humans. Surprisingly, no British dogs came down with mad cow disease, even though they were eating food very similar to the British cats. Any question about any of that? All right, let's briefly talk about plant viruses and viroids. Plant viruses are viruses that resemble animal viruses in many ways, and they cause many diseases of plants, especially diseases of economic importance, meaning crops such as beans, corn, sugarcane, and potatoes, I'm sure it happens to other plants as well, but we've studied mostly the economically important crops. Because of the tough cell wall, plant viruses enter the plant cells through either wounds or by parasites or by damage by insects. And I guess an insect can be considered a parasite to a plant. So nematodes, fungus, and insects tend to damage the plant and then make the plant susceptible to getting the plant virus. And usually the nematode or the fungus or the insect can spread the plant viruses as well as damage the plant. Infected plants can then spread the virus in their pollen or in their seeds, and then the plant virus continues. Any question about that?
Nope.
All right, we're not going to talk about plant viruses any further because plant viruses do not infect animal cells and they do not cause animal diseases. But you should know there are plant viruses and they do cause diseases in plants. Let me talk about viroids. Viroids are naked RNA molecules, and you can think of them as a naked RNA virus minus the protein coat. But they are infectious naked RNA molecules, and they can infect a plant cell and then cause plant diseases. Like I said, this is a naked RNA molecule. It doesn't have a protein coat around it. It is not a virus. Fortunately, there are no viroids known or found in animal cells, that there are only viroids in plants, and so they only cause disease in plants. The only one I'm going to mention is the potato spindle tuber viroid, which causes stunted potatoes. Let me see if I can find a picture of that. There we go. There's a picture of a normal potato, and these are stunted potatoes over here. Any question about any of that? All right, any questions at all?
Not so far.
All right. I think I want to move a little bit ahead. Actually, I'm not ahead. I got to talk about chapter 7 today anyways. Never mind. Yeah, let's move on to chapter 7. Hopefully I can finish chapter 7. It's a smaller lesson. This slide is telling you the rough outline and the major goals of chapter 7. Chapter 7 is an easy chapter for students to understand because it is, well, it's just a chapter that students already have a pretty good grasp on. It's talking about controlling microbial growth, things like disinfection and antiseptics. Students already have a pretty good handle and understanding of those, but know the terms of this chapter like all terms. 2, be able to discuss the factors that influence microbial death rates. 3, be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells. 4, understand the various types of disinfectants and how they work. And 5, understand the relative resistance of the major microbial groups to antimicrobial agents. Any questions about what we're going to cover?
No.
All right. When we're looking at the microbial death rate, bacterial populations die at a fairly constant rate. And if you graph it on a semi-log graph, meaning the y-axis is a log scale and the x-axis is just arithmetic scale of time, you'll note that on the log scale, the population dies, what's the word I'm trying to say? At a constant rate. If you were to graph it on a arithmetic scale, it doesn't quite die at a constant rate. It dies at a, I don't know, logarithmic rate, meaning there's more death at the beginning and then it starts tapering off. But when we look at that and we say that there's a million cells, after 1 minute, there will only be 100,000 cells remaining after treatment, meaning there's 900,000 cells that die. And then you do that for another time length, like 2 minutes, there will be only 10,000 cells alive. And then 3 minutes, 1,000 cells alive, meaning the number of cells is being decreased by a magnitude of 10. And the deaths happen by a magnitude of 10 also. Any question about any of that? So that means when you treat the population and there's 1 million cells and it dies with this scale, you know to get 1 cell, you need to expose the cells to the treatment for 6 minutes. Okay? Any question about any of that? All right. So the microbial death rate is affected by different treatments, and the effectiveness of the antimicrobial treatment will depend on a number of factors. One, I've already told you, the number of microbes present at the beginning of the treatment. The more microbes you have, the longer you need to treat to reduce to 1 or to none, which would be probably another minute. Because it wouldn't be quite 1, it'd be 1/10 of 1, but for all practical purposes, that's 0 cells. So the number of microbes present at the beginning of the treatment is depending on the microbial death rate. Environmental influences can affect the death rate also. The presence of organic matter can affect the treatment. Hospital specimens, let me add, usually have organic matter around them. Like when a patient has diarrhea and goes on the floor, or they throw up on the floor, that's organic matter in there. Organic matter may or tends to, depending on which treatment we're using, can inactivate the chemical treatment. For example, if the person is working with bleach, organic matter easily inactivates bleach. So the environmental influences can affect the treatment. And what you can do is, if you're cleaning up with bleach, you just use an excessive amount of bleach. The temperature of the treatment can affect the treatment. Generally speaking, the higher the treatment, the more effective the treatment will be, meaning the higher the temperature of the treatment, the better the treatment will be. The presence of biofilms can also affect the microbial death rate. Generally, if the bacteria has a biofilm, the cells will, or at least some of the cells deep in the biofilm, will survive longer from the treatment, and it'll be very difficult killing those cells. So the presence of biofilm means that the microbial death rate will be greatly changed, much less death rate. The time of exposure also affects the death rate. Obviously, if you have more cells, you need more time. You also need longer exposure for endospores or fungal spores, as opposed to bacteria, vegetative cells. You just need longer time for spores. Sometimes the treatment will not kill the spore, an endospore. The microbial characteristics can also influence the microbial death rate. I'm only going to talk right now about one, but if the bacteria have mycolic acid in their cell wall, and that will largely be the bacteria of the family Mycobacterium, sorry. Am I saying that right? Mycobacterium, that family. They have mycolic acid in their cell wall. That will prevent any water-soluble molecules moving into the cell. So bleach, for example, would not be a good treatment to use against the Mycobacterium. And that's a microbial characteristic affecting the microbial death rate. Any question about any of these? All right, am I on that slide? Yes. I guess I'll continue here. You should know the term sterilization refers to the destruction of all microbial life forms, including endospores. It may not include prions. Usually, sterilization is done by either steam or autoclaving, perhaps by ethylene oxide, a poisonous gas. And when we sterilize things in the lab, if you were to come into the microbial lab, we do that in the autoclave. Commercial sterilization refers to sufficient treatment, usually heat, to kill endospores of Clostridium botulinum in canned food. Disinfection just refers to the destruction of vegetative pathogens, not necessarily of the endospores. Antisepsis refers to the destruction of vegetative pathogens on living tissue. So if I use bleach on a toilet, I would call that a disinfectant. But if I were to use bleach on a cat, I would call that use an antiseptic. Any question there? Degerming is the removal of microbes from a limited area, such as degerming the skin before an injection site. So when they do an alcohol swab of your skin before injecting you, they're not obviously doing sterilization. They're not even doing disinfection. All they're doing is degerming, reducing the number of microbes in that area. Okay? Sanitization is the treatment intended to lower microbial counts on eating and drinking utensils to save public health levels, and this is usually done in someplace serving food, and they sanitize their eating utensils before serving the food. Any question about any of that? All right. Some other terms you should know. A biocide or a germicide is a substance that kills microbes. It does not usually kill endospores. Bactericides is a substance that kills bacteria. Fungicides, a treatment or substance that kills fungi. Virucides, a treatment or substance that inactivates viruses. biocide kills bacteriostasis, inhibits the growth of microbes, but does not kill the microbe. And I believe we talked about this in the lab. You can have an antibiotic, which is bioacidic. What do we call that? Bio antibiotic. I don't remember what we called it. Or bacteriostatic. A bacteriostatic antibiotic only inhibits the growth of the microbes. It does not kill them. Once you remove the bacteriostatic agent, the growth might resume. With a bacteriostatic antibiotic, what we are doing is preventing the bacteria from growing and then allowing the host immune system to remove the microscopic agent. And so you need the patient's immune system to remove the bacteria. Any question about any of that? Sepsis refers to microbial contamination. Asepsis is the absence of contamination by unwanted organisms. Aseptic techniques are important in surgery to minimize contamination from the air, from the patient, their own skin or whatever is being operated on, as well as the surgical tools and obviously the surgeon. Any question about any of that? All right. Antimicrobial agents kill or inhibit microbes by one of three ways. They can first alter the membrane permeability. You damage the plasma membrane, either the lipids or the proteins. This will cause the cellular contents to escape from the cell, interfere with normal metabolism, and kill the cell. You cannot lose the cytoplasm of the cell and retain the cell's living status. If there's a hole in the cell membrane, cytoplasm leaks out, that will kill the cell. Oh gosh, I just forgot the name of that antibiotic that actually kills cells that way. Something B, polymyxin B is an antibiotic that kills cells by altering the membrane permeability. You can also damage intracellular proteins. You denature proteins, the enzymes, by targeting disulfide bridges or denaturing the proteins in some other ways, like damaging their hydrogen bonds. That will cause cell death. Alcohol can damage cells by denaturing proteins, although alcohol can also destroy the cell membrane and kill the cells that way. Penicillin G damages peptidoglycan. That's not exactly a protein, but it does have peptides in it. And that's one way that penicillin G works. Not exactly a protein, but close to a protein. It's a peptide. You can also damage nucleic acid. This often results in cell death. Whenever you damage the nucleic acid, you cause a mutation. And 5 or 6 mutations is all a cell can handle, and then the cell will die. Ionizing radiation tends to damage the cell by damaging the cell's nucleic acids. The same with UV light. Any question about any of that? All right. There are physical methods we can use to control microbial growth, or we can use physical methods to kill microbes. And you can use heat, filtration, low temperature, high pressure, desiccation, osmotic pressure, and radiation. These are all physical methods. Let's talk about some of them. Heat is a physical method. All methods of heat kill microorganisms by denaturing proteins and enzymes. Though if you use flaming and incineration, that's very high heat, you actually turn the cell into char. And that, of course, kills the cell too. You have two different types of heat. There's wet heat and dry heat. Wet heat includes boiling, autoclaving, and steaming. Dry heat includes baking, like cooking. It also includes flaming and incineration. Realize this is much higher temperatures than baking, or much higher temperatures than boiling or autoclaving or steaming. I don't know what flaming is, but it's very high and it could get up to 1,000 degrees. I don't think it has to get that high, but it's not a temperature you cook at. Okay. Moist heat denatures proteins and enzymes. Boiling, that's 100 degrees Celsius, boiling liquids or flowing steam, kills vegetative bacterial pathogens, most viruses, fungi, fungal spores, and all you need is 10 minutes to kill vegetative pathogens. Most of the time, the cells will die much faster. Like for E. coli, all you need to do is bring the temperature to boiling and then take it off the boiling, and the E. coli is dead. Endospores and some viruses do take longer to destroy. Some endospores can survive 20 hours in boiling water. Endospores are very difficult to kill. There are only a few treatments that can kill endospores, and moist heat is not one of them. An exception is autoclaving. I don't know why, but the temperature and the pressure of autoclaving is enough to kill an endospore for the volume we use, which would generally be 1 liter. If you were to use more than that, you may not kill all the endospores, but you could use longer. If you had larger volume, you could use a longer autoclave time. So reliable sterilization by moist heat requires temperature above boiling. How is this achieved? By autoclaving. Autoclaving, Steve, under pressure at 121 degrees Celsius. The pressure is 15 pounds per square inch. Sterilization works very well. The pressure does elevate the boiling temperature, why the liquids can get to 121 degrees, because it's under pressure. You can also use steam, but steam is good at sterilizing surfaces. It's not good if the steam cannot directly contact the microbe. Like if you had, I don't know, a container and the container is closed and you wanted to sterilize the inside, you would not be able to sterilize the inside of it with steam, because steam has to come into contact with the microbe. All right, another moist heat is pasteurization. Pasteurization does not kill all microbes. All it does is it reduces the number of spoilage organisms, and it does kill all pathogens. It's mild heating, sufficient to kill organisms that cause spoilage. The mechanism is denaturation of proteins and enzymes, but thermoduric organisms can survive pasteurization. There are two forms of pasteurization. treating at 63 degrees Celsius for 30 minutes or 72 degrees Celsius for 15 seconds. Most pasteurized milk is sterilized at 72 degrees Celsius for 15 seconds. Pasteurized milk does have microbes in it. That's why it has a shelf life. It usually has an extended shelf life compared to unpasteurized milk because the number of microbes in the milk is reduced, but they're not eliminated. The milk will go bad, and that's why you should throw it out shortly after the expiration date, which is actually the date the milk must be sold by. It's not the date the milk must be used by. And what you can do is just keep the milk until it starts to curdle or go sour, and then you can throw it out. Although my mother would actually eat the curdled milk. I don't recommend that, by the way, because the wrong organism might be growing in it. Milk can also be sterilized at 140 degrees Celsius for about 1 second. This is not pasteurized milk. It's called ultra high temperature treated milk, or UHT milk, and that sterilized the milk. This milk does not require refrigeration until it's open, and you can see it in the store in a package kept at room temperature. And that's usually what they'll have with milk, although they could irradiate it too, in which case it's called irradiated milk. And it can be kept in an open package at room temperature as well, because it's sterilized. Any questions about wet heat? If not, let's move on to dry heat. This picture is to show you that dry heat is not as effective at killing as wet heat, because this cat climbed into the oven to get warm. Okay, so dry heat sterilization is not as efficient as wet heat sterilization. And the cat's proving that. Dry heat kills by oxidation. Some examples are flaming and incineration, which is not shown here because the cat would be char. But hot air sterilization, more commonly called baking. To sterilize something with dry heat sterilization called baking, you need to place it in the oven at 107 degrees Celsius for 2 hours. If you remember, placing in boiling water, all you needed to do was put it in 10 minutes. So wet heat is more efficient than dry heat. And you actually know this because everybody sticks their hands into a hot oven to bring out whatever is in the oven. That does not burn your hand. If you were to stick your hand through steam, that would burn you immediately. So you can't stick your hand through steam or in boiling water, but you can stick it in a hot oven. You can also control microbes by filtration. Filtration removes microbes from liquids and gases. You put whatever is being filtered through a filter that has pores, and the pores allow the medium to go through, either the liquid or the gas, but not the microbe. It's useful for sterilizing liquids that would be altered by heating. Some examples of juices, Coors beer, vaccines. Milk is not usually filtered because the fats and the protein would clog the pores in the filter. Coors beer is filtered, and so some people like it because it has a different taste than pasteurized beer. Some juices are filtered because we want to keep the original taste of the juice. the original vitamins, the original color, and so they filter the juice. Any question about any of that? All right. You can also put microbes in low temperature. Low temperature does not tend to kill, at least not all microbes, but it does inhibit microbial growth. Refrigeration, 0 degrees Celsius to 7 degrees Celsius, slows the metabolic rate of most microbes. It does not prevent the growth of all microbes. There are some microbes that can grow at this temperature, but they grow slowly. Mesophiles generally cannot grow at this temperature. And it's usually mesophiles that cause human disease. It's actually all of them that cause human disease. But it's either a mesophile or a cold growth organism that causes the food to spoilage. And that's why after 1 week, the microbes that grow at this temperature can grow to such numbers to cause the food to spoil. So if the food does not have another preservative in it, like a sugar or a salt, the food should be thrown away after a week in the refrigerator. A psychotroph is an organism that can grow at this temperature, and after a week, 1 cell can grow to 2 million, causing spoilage. Freezing, this is temperatures less than 0 degrees Celsius, down to, I don't know, -10, -20 degrees, stops metabolic activity. Slow freezing in your home freezer allows ice crystals to form, which kill some bacteria, but not all. About 50% of bacteria will survive freezing and thawing, but the freezing does kill all parasitic species, like parasitic worms and parasitic eukaryotes, the protists. So if you wanted to make sure your meat did not have trichinosis or something like that, you put it in the freezer. That isn't true for quick freezing. When you put something in liquid nitrogen, it'll freeze so quickly, the ice crystals do not form, and that would not kill the bacteria or the eukaryotic parasites. Quick freezing was putting something in liquid nitrogen. And I actually preserve bacteria in liquid nitrogen. High pressure transfers pressure evenly throughout a liquid, killing vegetative bacteria cells. It does not kill endospores, but the vegetative bacteria will be the most numerous. And depending on what the species is, it may not have endospores. High pressure denatures proteins, alters carbohydrates. Like I said, endospores are resistant to high pressure. We use high pressure treatment to preserve the color, the flavor, and the nutrient value of the food, like the vitamin C, would not be affected by high pressure. Juices may be treated by high pressure. Heating would remove vitamin C and some nutrients, and it may change the flavor. It may change the color too. This is a joke showing you the son or daughter, the child, and the mother, their lumps of coal. And the mother says, Dad's been under a lot of pressure lately. And the pressure changed Dad into the diamond. Desiccation and osmotic pressure have been used by humans to control microbial growth for centuries, maybe even millennia. Desiccation is drying. This prevents microbial metabolism. They need water around to have normal metabolism. Some cells can remain viable for years, like endospores. The resistance to desiccation varies by species, like E. coli. As soon as you dry it, the E. coli will shortly die. Examples of desiccation: drying fruit, like dried figs, osmotic pressure, molasses, honey. All of them are using desiccation and osmotic pressure to preserve the food. If you don't know, the molasses can be kept at room temperature for years. My mother had a bottle of molasses that we figured was over 30 years old before my mother decided, maybe I should throw that away. And neither I or her were ever using it. So I said, yeah, go ahead. I did use it when it was about 20 years old. I made molasses candy for my father, and he loved it. It didn't kill him, and it was 20 years old. So the molasses had osmotic pressure to prevent the microbe from falling in and then causing food spoilage. High osmotic pressure is usually happening by having high salt content or a high sugar content, causing plasmolysis, where liquids will leave the bacteria. The plasma membrane will pull away from the cell wall, and that will typically kill the bacteria, at least the vegetative bacteria. Some examples, like I said, molasses and honey. Something that has all three: high salt, high sugar content, and then a high acid content, is ketchup. And then ketchup is usually kept in the refrigerator, so that increases shelf life too. And your bottle of ketchup can last for years. I've got a bottle of ketchup that's at least 3 years old. Tells you I don't go through ketchup very quickly. Okay, and it's still good because I haven't used it for, well, I don't know, a couple of weeks, but the last time I used it, it was still good. I didn't die. All right. Another physical method for controlling microbial growth is radiation. Radiation damages DNA. Ionizing radiation has radiation with high energy, like X-rays, gamma rays, electron beams. They have a short wavelength, high energy. When the energy hits a molecule or atom, it will give the electron, which absorbs that energy, so much energy, the electron will leave the atom or molecule, which will create an ion, meaning an ion is a atom or molecule which has a charge. When the electron leaves, that will give a positive charge to that atom or molecule. And that's why we say it creates ions. The ions tend to bump into other molecules and steal electrons. And when they do that, they can damage the other molecule, such as bumping into DNA, stealing the electrons from DNA, damaging the DNA, and causing mutations. Ionizing radiation can also cause hydroxyl radicals, which essentially do the same thing. They can bump into different molecules and damage them, stealing electrons from DNA or another molecule, damaging them. Ionizing radiation is sometimes used in the food industry, pharmaceuticals, and medical supplies to sterilize the food and then keep it at room temperature and seal to protect it from microbes. Any questions about any of that?
Nope.
Great. Non-ionizing radiation is, actually, we're talking about ultraviolet light. Non-ionizing radiation is actually ultraviolet light, visible light, and one other form of light near visible light, and I forget what that is. If it's microwaves or if it's heat, I don't remember. But ultraviolet light is the non-ionizing radiation that damages DNA. When ultraviolet light hits DNA, what it does is it causes two adjacent thymines damage where the two adjacent thymines form a dimer where they are covalently bound to each other. That's called the thymine dimer. And these two thymines do not hydrogen bond to their corresponding DNA, the A. Normally, thymine does not bind to the other base near it, even if it's a thymine. But when you expose two thymines, has to be two thymines together, they'll get a thymine dimer mutating the DNA. This portion here, the DNA forms a bubble, and the polymerase cannot replicate this, nor can the RNA polymerase transcribe this. So that's how it mutates the DNA. Microwave radiation is a form of non-ionizing radiation. It is particularly not antimicrobial, but microwave radiation tends to cause heat, and then the heat tends to kill the microbes. So microwaves are not particularly antimicrobial, but they do cause heat, and then the heat can kill the microbe. All right, this table is showing you the summary of the methods of microbial control. I want to move quickly through this, so I'm just going to mention it. And I'm going to tell you to study the summary. We talked about it already. Let's briefly talk about the chemical methods of microbial control. Previously, we talked about physical methods where we control microbes with various chemicals. There are few chemical agents that effuse sterility. Most chemical agents just decrease microbial numbers to safe level, and most remove vegetative forms of the pathogens. They do not necessarily remove the endospores. There are some principles of effective disinfection. Disinfection is the destruction of vegetative pathogens by a chemical agent. There are different factors related to effective disinfection. The concentration of disinfectant, generally speaking, the higher the concentration, the more effective it'll be. The time and access to the surface of the disinfectant, meaning the disinfectant has to come into contact with the microbe. And generally, the more time you have, the greater the disinfection. The pH, generally, the more acidic the pH, the better the disinfectant will work. There are some chemicals where the greater the alkalinity, the greater the disinfection. But generally, more acidic pH, the greater the disinfection. The presence of organic matter tends to inactivate many chemical disinfections. And remember, organic matter is often found in clinical situations. So you want absence of organic matter to improve the chemical disinfection. Here are the different types of disinfections, disinfective agents, the chemical disinfections, and these are just categories. Let me talk briefly about this. Phenol, phenolics, biphenols are compounds based upon the chemical phenol. You don't need to know the mechanism of action and the preferred use. The table is telling you you need to know the different agents and the preferred use. Phenol is actually hardly ever used today because it is fairly corrosive to metals, is fairly irritating to the human skin. But this is what Joseph Lister used to perform his aseptic surgery, because in his day, phenol was the strongest antimicrobial chemical disinfectant. Today, we have many better agents, though. Biphenyls are in disinfectant hand soaps, skin lotions. Phenolics, we sometimes use them to treat instruments, skin surfaces, mucous membranes. Bigonides, skin disinfections, especially for surgical scrubs like chlorhexidine. Halogens, iodine is actually something that used to be used for a surgical scrub. We have a slightly better agents now, but it used to be a very good antiseptic agent and used all the time in surgery. Chlorine bleach is another halogen, which we use as a disinfectant to clean things like toilets. Alcohols, these are very weak chemical disinfectants. They're so weak, we call them degerming agent, as opposed to a true disinfectant. Heavy metals like silver nitrate fell out of use for a while because they're somewhat toxic to humans. They're not that effective. And there were better agents. However, because of the resistance of microbes to especially antibiotics as well as other antimicrobial chemical agents, their use has come back, and they're used in things like surgical dressing, where they're put on the dressing to prevent the growth of microbes in a wound. Soaps and detergents are mostly skin degerming agents. They are not very good at killing cells. They only killed about 10% of bacteria on your hands, but they do wash away microbes, and we call them degerming agents. Sanitizers, antiseptics for the skin fall within the surface active agents. Chemical food preservatives, these are preservatives we add to food to keep the shelf life longer of the food. Meat products such as ham, bacon, hot dogs contain things like nitrates and nitrites. And then sorbic acid, benzoic acid can be put in various food like bread, calcium propionate put into bread. They can put into other agents too, like cosmetics and shampoos. And that's just to keep the agent from getting microbes to grow in it. Aldehydes, glutaraldehydes, formaldehyde, used as a disinfecting agent, like disinfecting microbial equipment, medical equipment, because they're not corrosive to metals. Ethylene oxide is a chemical sterilization of poisonous gas. It's very effective. However, it requires long-term exposure, and ethylene oxide is very toxic to humans. So you need to put it in a closed environment, make sure it doesn't get out and poison someone. It can kill endospores, but you need long-term exposure. It's sometimes used for sterilizing medical equipment, anything that could be damaged by heat or a corrosive agent. Sometimes you use ethylene oxide. Peroxygen, such as hydrogen peroxide, is something that can kill endospores, and it is a very good antimicrobial or antibacterial agent. And it is one of the few agents that can kill endospores. Like I said, ethylene oxide can, but you need long-term exposure to kill endospores, With hydrogen peroxide, you do not need long-term exposure to kill endospores. So this is one of the few treatments that can kill endospores, and it's very effective at killing endospores. All right, comparing the effectiveness of various antiseptics, the weakest that I'm showing is soap and water. Nobody washes their hands for 2 minutes, but even if you did, you would kill, what is that? Only about 90% of the, maybe 80% of the microbes. Most people only wash their hand for maybe 30 seconds. And so you're only killing about 5 to 2% of the microbes. And then something I want to point out, tincture of iodine. That was the agent I told you they used to use for surgery all the time because you coat the skin with it, meaning it stays on the skin for a long time. And after only 80 seconds, you have 10% of the microbes still alive. And obviously, when you put it on the skin, it stays there for a long period of time. Any question about any of that? All right. Microbial characteristics can affect microbial control. We can make a hierarchy of what's the most resistant and what's the least resistant. And this is a simplified hierarchy of the different microbial groups to the chemical biocides in general. The most resistant microbe is a prion. Essentially, there's no chemical agent that can destroy a prion. To kill prions, you really need to take it to 1,000 degrees Fahrenheit and incinerate it. And that's not a chemical agent, that's a physical agent. There is one chemical agent that does work, and it's experimental. You can use proteinases. Proteinases destroy the protein of the prion, which can remove the prions. Endospores of bacteria are the next most resistant agent. Mycobacteria, the next most resistant. The mycolic acid prevents any bacterial static agent, chemical agent, that is water soluble from entering the cell and then damaging the cell. The cystiprotozoa, the next most resistant. Vegetative protozoa, the next most resistant. Gram-negative bacteria, the next most resistant. Having that gram-negative, the outer membrane of the cell wall, makes bacteria more resistant than a gram-positive bacteria to many antimicrobial agents, chemical agents. Fungi, including most fungal spores, the next most. Viruses without envelopes, the next most resistant. Gram-positive bacteria, the next most resistant, which would be the second least resistant. They're the second easiest group to kill. And then viruses with lipid envelopes, the easiest group to kill. You damage either the protein, the lipid, or the genome of the virus, you damage the virus. Any questions about any of that?
Nope.
All right, I talked about that. All right, I mentioned a little bit of mycobacteria having greater resistance because of the mycolic acid. Endospores, very few biocides, chemical agents that actually work on endospores. Hydrogen peroxide is one, and that is found in almost all households. About viruses. So let's move on. I did mention prions. How do you kill a prion? It's not completely solved yet. Like I said, the British, what they did was they incinerated all of their cattle to kill the prions. That is one way. And it's not clear why the prion protein needs to be incinerated to inactivate it. Because most proteins, once you heat them, they become coagulated, or what's the word I'm looking for? And that doesn't seem to work with prion proteins. You need to actually incinerate them. But you can treat them with proteinases, and that digests the proteins, destroying the prions. All right, let me go previous. Let me talk a little bit about the mycobacteria. The mycobacteria have mycolic acid in their cell wall, so they are very resistant to most chemical agents like bleach or hydrogen peroxide. I think hydrogen peroxide. I'm not sure if that's lipid soluble or not, but I think it is, in which case it would not work against the mycobacteria. However, something that can kill the mycobacteria, which is a very poor agent, is actually alcohol. Treating the mycobacteria with alcohol. The alcohol goes right through the mycolic acid and then denatures the protein and the cell membrane of the mycobacteria and can easily kill the mycobacteria. So this is one agent which is a really poor disinfectant. That's why we call it a what do we call that? A degerming agent. It's so poor of disinfectant, we don't call it a disinfectant. We call it a degerming agent, but it's actually very good at killing the mycobacteria. And this is one characteristic of this group, which makes it sensitive to alcohol, but it is resistant to most other chemical disinfectants. Any question about any of that? All right. While microbial control methods are effective, they, especially the biocides, are not uniformly effective against all microbes. The compounds discussed in this lecture are not generally useful in disease treatment, with the exception of the antibiotics. Any question about any of that? All right. Sorry I went a little bit long for this lesson, but I wanted to finish it so I wouldn't have to talk about it in our next lesson. In our next lesson, we will cover a review for the final. Any questions about any of this?
Nope.
All right. Since it's after 6:30, I'm not going to have a lab for today. Okay. So I'll show up just in case anyone's there, but if they're not, I'm going to log out. So if you have a question, you're going to need to email me.
Okay,
All
so
right.
no lab.
No lab.
Okay,
All right.
thanks.
All right. I'm going to log off and I'll say I'll see you next time.
Okay, bye.
All right. Bye.